Welcome to this lecture number 20 for the course. We're going to talk today about carbohydrate biosynthesis as well as steroid biosynthesis. There are four major types of carbohydrate biosynthesis we're going to talk about today. We're going to talk about the conversion of CO2 to glucose in plants. That's very brief, and it's going to be very much a review. So is the next section, which is on gluconeogenesis, or the synthesis of glucose in animals and humans. So both those should be either really top level overview or review. The two new topics are glycogen synthesis, which is making of glycogen, and then also the conversion of glucose to other carbohydrates, in particular how that relates to the process of post-translational modifications of proteins. In animals, our blood glucose and our glycogen are our main carbohydrate biosynthesis mechanisms. Shared between both animals and plants are glycoproteins, disaccharides, and other monosaccharides, and exclusively in plants we have our starches and our sucroses. So we're mainly going to be talking about this area of things, although we'll briefly introduce the plant topic. Again, as a general reminder, gluconeogenesis is the manufacturing of glucose from our carbon structures that are at the end of glycolysis, so things like our pyruvate, phosphoenol pyruvate. And as a general reminder, gluconeogenesis is very similar to glycolysis. In fact, 7 of the 10 enzymes are the same, and they just run in reverse. So, and if you want to reference that, there's an excellent lecture that was given on that topic last week that was on the same lecture as glycolysis. Again, as another quick reminder, ribose and ribulose, which are 5-carbon carbohydrates, those are made in this pentose phosphate pathway, which sits right next to glycolysis. Fructose 6-phosphate gets converted into xylulose, which can get converted into ribose and ribulose. So the pentose phosphate pathway serves as a generator of 5-carbon carbohydrates. That's important in nucleotide biosynthesis. Also sitting next to glycolysis is glycogen metabolism, and if we take glucose 6-phosphate, we can make glycogen, which is, again, a storage molecule, which we'll talk about here in a second.
The other main way is photosynthesis, and there's two types of photosynthetic reactions. One is a light reaction, where we can convert solar energy, which is sunlight, to chemical energy, both ATP and NADPH. And there's also dark reactions, also known as the Calvin cycle or Calvin and Benson cycle. These are light independent reactions, but they use the energy produced and stored from the light reactions to generate and incorporate CO2 into an organic molecule called pGal, which is later converted into sugar. That's the general gist. So there are two ways we do this. Again, light is going to convert into ATP and NADPH, and from there we take the Calvin cycle and we can generate and spin off all our sugars, and we can also spin off and we spin off and take those reactants and put them back in so they can be recycled so we can generate more sugar. So we're taking CO2, putting it in a Calvin cycle, and we get out sugar.
So the net reaction out of photosynthesis is 6 water, 6 carbon dioxide. We're going to add in energy in the form of sunlight at the site of chlorophyll, and from there we're going to generate 6 glucose molecules, which then can be used in glycolysis. And we're also going to generate oxygen, which is also very beneficial. So that's the general pathway. So now next we'll talk about the light and the dark cycle associated with photosynthesis.
Okay, so the first process in photosynthesis is the light reactions. The light reactions are called non-cyclic photophosphorylation. Photo because you're going to take light, phosphorylation because we're going to make ATP out of it. And so electrons are removed from water and they're passed through this system in order to generate that energy. And basically the steps here, so light's absorbed in this PS2 system. They're passed from pigment to pigment until they reach a molecule called P680, and from that high energy electron is passed to an acceptor molecule and replaced with an electron from water. So we split water and we get O2 in the end. The high energy electron then travels down the electron transport chain. It does lose energy as it goes. Some of the release energy drives the pumping of protons, and it builds a gradient. And again, in step 3, we're going to have another exception of a proton. Ferredoxin is then transferred. And from there, we're going to have a pump that's going to, again, generate ATP, just like the FOFA F1 ATPase does, or I should say, in a very similar way to the FOF1 ATPase, but we're basically using electrons to be transported off in order to create a pump and to create NADPH and that proton gradient. The next is the dark cycle. So dark cycle is also called the Calvin cycle. It uses energy from the previous step to create CO2 and pGal. What you basically have, again, CO2 is then converted into glucose. So in an enzyme that's abbreviated Rubisco and ribulose bisphosphate, which is labeled RUBP, have 5 atoms of carbon and a phosphate group on each end. So Rubisco catalyzes a reaction between carbon dioxide and ribulose bisphosphate, which then forms a 6-carbon compound that is immediately converted into 2 3-carbon compounds. And this process is called carbon fixation because the CO2 is fixed from an inorganic CO2 molecule into an organic molecule. And ATP and NADPH use their stored energy, and that's how they're converting those the 3-carbon compound 3-PGA into another 3-carbon compound called G3P. And this type of reaction is a reduction reaction because there's a gain of electrons. So a reduction in the gain of an electron by an atom or molecule is the story there. The molecules of ADP and NADP are result, and they are returned to the light dependent. So they return to the light reactions to be re-energized. One of the G3P molecules leaves the Calvin cycle to contribute to the carbohydrate molecule. So that's commonly glucose, and so that's right after the second stage. That's where that happens. So the carbohydrate molecule has 6 carbon atoms, and it takes 6 turns of the carbon cycle to make 1 carbohydrate molecule. So 1 for each carbon dioxide molecule fixed, and the remaining G3P molecules regenerate RUBP, which enables the system to prepare for a carbon fixation step.
So that's the photosynthetic pathway very briefly. Next, we're talking about glycogen. And glycogen is where we're going to add glucose in a multi-branch polysaccharide. So the structure is shown here at the bottom here, where we have multiple glucose molecules in a long chain and also in a branched chain. So this is the summary of this step. Basically, we're going to go from glucose 1-phosphate down through we're going to add a UTP to it to get a UDP glucose, and then we're going to add the UDP to glycogen. So step 1, we're going to take our D6 glucose 6-phosphate from glycolysis, and we're going to basically move that phosphate group from the 6th carbon, down to the 1st position. And that allows us then to add the UDP or UTP to the glucose 1-phosphate and get UDP glucose. So that becomes the activated glucose donor, and UDP glucose phosphorylase is the enzyme that catalyzes this reaction. UDP glucose, this is the flat structure of that. You can see that there's a glucose molecule on the top in pink. There are 2 phosphate linkages, and from there we have our nucleotide. It's uridine. So that's the common structure. That's the end result of step 2. In step 3, we're going to take that UDP glucose, and we're going to add it to the non-reducing end of a glycogen molecule. And that is going to add basically 1 more unit to the glycogen molecule, and we're going to kick off a UDP. So we're just basically making these points. We do have the ability to branch off this, so the common addition point on glycogen is alpha-1,4, but we can also branch at an alpha-1,6 using a branching enzyme, and the synthases will then just keep extending both of those, and we end up with a very complex branched carbohydrate, glucogen. And this is kind of what the structure looks like, and you can, in both representations, it gives a very clear conformational identity, and you can kind of get an idea of how the branching works. Regulation of this is through a couple different mechanisms. The glycogen is phosphorylated. It's in the inactive form, and it states in its serine/hydroxyl group in the activated form. To activate it, okay, if we're going to activate it, insulin will activate it, glucose 6-phosphate will activate it, and glucose will activate it. Glucagon will inhibit the activation, glucagon and epinephrine. Insulin will also deactivate the inactivation of the phosphorylation of this glycogen synthase B. So that's kind of how everything's regulated. It's all regulated by feedback mechanisms and other hormonal regulations. And that's the insulin, basically, this is the pathway by which it does that. So insulin binds to the insulin receptor. That was one of the molecules we 3D printed. We go through one of our biosignaling pathways through PI3K, through our PD kinase 1, and phosphokinase PKB, and that's going to then interface with GSK3, which will inactivate GSK, and that will allow for the activation of glycogen synthase. That's the role of insulin in this whole process. And that kind of makes sense because insulin is one of the controlling mechanisms of carbohydrates/glucose. This just kind of gives you an idea of how low blood glucose or high blood glucose leads to glycogen synthesis, upregulation or downregulation. Just a flow chart, and shows you how all of that goes together and in what pathway. I'll use it for your reference. You don't need to necessarily memorize this. It's just more of a reference tool. The last topic in carbohydrate synthesis is oligosaccharide synthesis. And basically, as a reminder, we basically take proteins that are embedded in a membrane via various mechanisms, and we're going to add carbohydrate structures to these proteins. And we can create some very complex and complicated structures using these carbohydrate additions to protein. So if we're going to generate some of these new 6-carbon structures, such as fructose, mannose, galactose, there's several pathways that we have. From glucose, we can make fructose using our glucose 6-phosphate. From mannose to mannose, phosphorylated 6-carbon. Mannose, we're going to use GDP mannose phosphorylase and convert it to the first position, and so on and so forth. The ones we don't have are galactose, and that is just an uncharacterized enzyme that we have at this point, but this is how we generally are going to go, an example of how we're going to go from a known entity in the cycles to an unknown entity in the cycles, and that allows us to then, through enzymes like mannose, we're going to add these proteins on. So we have a high mannose sugar that's going to be added by mannose. We can also add, if we're going to add a glucanac, there's a glucanac transferase, and other various different enzymes that allow us to create complex carbohydrate structures on the top of these proteins. So that is carbohydrate biosynthesis in a nutshell. So the next step of this process, we're going to talk about steroid hormone biosynthesis. As a reminder, there are 4 classifications of hormones. There are protein and peptide hormones. We're not going to talk about any of those today. There's steroid hormones. Those are the main ones we're going to talk about today. Cortisol, aldosterone, estrogen, androgens, progestogens, and vitamin D. And then we'll spend a little bit of time talking about, we're not going to talk about amino acid derivatives too much. We will talk about adrenaline very, very briefly, and we'll talk about our prostaglandins, our thromboxanes, and our prostacyclins, very, very briefly as well. So those are the four major categories, and those are what we're going to talk about. We're mainly going to talk about steroid hormones that are derived from cholesterol. We'll also mention the other ones. So as a general reminder, there's mechanisms of hormone action. We have two main ones. First, peptide hormones and catecholamines can bind to a cell surface receptor. That's what you see on the left. A peptide hormone comes in, binds to that cell receptor, and then we're going to get upregulation of transcription. And so you see that in step 4 and 5, and from there an mRNA is produced, and from there you get new proteins. So that's the first mechanism. The second mechanism on the right is steroid and thyroid hormone. Steroid and hormones are circulating in the circulatory system. They pass through a cell membrane. Generally, this is just a passive diffusion. Then they're going to complex with another protein. That complex is going to allow the molecule the hormone and the binding protein to act as a transcription factor, an upregulator, and so then we end up with more protein transcribed. So that's the second one. I should say more proteins generated. As a general reminder, again, steroid hormones are all based on cholesterol. They're all started from cholesterol, and so all steroids are lipophilic molecules, and all the steroids except for calcitrol have cyclopentopyrhydrophenylanthrene structure, which is also referred to as a styrene. So this is the precursor molecule for all of those steroids. There are 6 main steroids class. In yellow, we have our prostaglandins. In blue, we have our androgens. In purple, we have our mineralocorticoids. We're not going to we'll discuss those very briefly, but not in any great detail. In green, we have our glucocorticoids. We also have some of our vitamin D, which are not shown in this chart, and some of our estrogens are in here, but they're also inside this triangle right here. So this is the estrogen class right here. Steroid hormones, these are the major ones. This is a generic pathway and shows you how you make different hormones from different precursors. Obviously, you can see cholesterol is the major. It's the start. All other hormones are generated from it. Progenolone is an intermediate for most of these as well. Enzymes that produce the steroids from cholesterol are located in the mitochondria and the smooth endoplasmic reticulum. And since steroids are lipid soluble, they're freely able to permeate the membranes and they're not then stored. And that's really good because they're an important signaling molecule and they need to be in the concentrations that are physiologically relevant. This is a very detailed chart of all the hormones and the enzymes that exist to generate those molecules and the pathways. I'm not going to go in detail on all of them. I'll go in detail on some of them, but this is a good reference tool for you. Again, we start with cholesterol, and we are going to go to pregnenolone, and from there we can branch off to all of the other steroid molecules. As a general reminder, steroid molecules are found in a complex. Sometimes they will just pass through by passive diffusion through the outside membrane, bind to a carrier, and then go to a cellular receptor, in which case they then are upregulating gene expression commonly in the cell. Here's a nice little chart. Cholesterol goes to progestins. Progestins then can be made to make glucocorticoids, mineralocorticoids, androgens, and androgens then can be made to estrogens. So these are all of the steroid hormones and how they're envisioned. Pregnenolone belongs to a group of neurosteroids, and they're found in high concentrations in brain, and they're synthesized there. They affect synthetic functions. You can see that from cholesterol, we're basically going to use the enzyme mitochondrial P450-SCC. So it's the rate-limiting step in this steroid hormone synthesis, basically all steroid hormone synthesis, and it's important this molecule is an important precursor of all C18, 19, and 21 steroids. And so this is an important, not just directly as a hormone, but it also serves as a precursor for a large chunk of the other steroid hormones. Progesterone, it's a progestin. It's produced directly from the pregnenolone. It's secreted from the corpus luteum. One of the differentiating factors in mammals, and the conversion of the pregnenolone to progesterone takes place in two steps. First, we have a 3-beta hydroxyl group is oxidized to a keto group and moved to C4, and this reaction is catalyzed by 3-beta hydroxysteroid dehydrogenase and isomerase. So this is the reaction that occurs in order to make this progesterone molecule. Steroids of the adrenal gland include cortisol, corticosterone, aldosterone, and androstenedione. From there, again, we are all going through cholesterol, through pregnenolone, and you can see all the enzymes and how those are made. Cortisol is a dominant glucocorticoid in humans on the far left, and it's synthesized from progesterone in the zona fasciculata in the adrenal cortex. It's adapted stress. It's adapted through stress. It's slightly elevated in the liver and has numerous effects on the immune system and certain T cell. And androstenedione is a principal mineral corticoid, and it's produced from progesterone in the zona glomerulosa of the adrenal cortex. It occurs at high level during stress and raises blood pressure and fluid volume, and it's an important element in many other cycles. Okay, we're going to keep going. Those are the two other classes that we had mentioned earlier. Two of the other classes we had mentioned earlier. From here, we've already talked about cholesterol, progesterone, and androstenedione. From here, we're going to talk about basically estrogen and testosterone. As an intermediate between pregnenolone and androstenedione, we have this dihydroepipenandrosterone. It's a weak androgen that can be easily converted into estrogen. You can see that in the chart. It has various protective effects, and it plays an important role in aging and is regulated by NAD coenzymes. So it's kind of an intermediate. It doesn't have too much of a direct effect. It's mainly a transient effect, and it's mainly used as an intermediate for other steroid-generated biosynthesis. Testosterone, that is the next step from androstenedione. It's converted from commonly referred to as DHT or dihydroxytestosterone as an intermediate. It's produced in the sperm in Sertoli cells, and it's very, very important, obviously, in secondary male characteristics, and it serves as an intermediate from progesterone. Estradiol, again, comes from testosterone. It's a female hormone. It regulates gonadotropin in the ovarian cycles, and with progesterone, estrogen is a uterine endometriosis maintenance factor. It's a differentiation of a mammalian gland, meaning that's an important hormone in a mammalian species, and it's responsible. This molecule is responsible for the secondary female sex characteristics and other. It actually has numerous mechanisms that we're not necessarily fully aware of. Again, hormone stimulation depends on the cell type and the receptors. So for cortisol synthesis, we have ACTH. For estradiol, we have FSH, follicle-stimulating hormone. For LH, which is luteinizing hormone, we have testosterone synthesis. So it depends on the cell type and the receptors that are available. And hormones bind to cell membrane these hormone cell receptors, in this case, and are activating through various biosignaling pathways, including G proteins, calcium channels, phosphatidylcholine, inositol mechanisms, and we're going to then basically upregulate expression. So that's basically how our steroid hormones work. and all the ones basically we've talked about. So generally speaking, when we're talking about biosynthesis of steroid hormones, this is a critical step in cell activity in mobilizing cholesterol from their cholesterol from HDL, which we talked about earlier. So we're going to convert those, we're going to transport those cholesterols to the mitochondrion. And again, cholesterol is generally referred to as that small molecule that is found in circulating fatty acids. That's a general step, but really it's that hormone. So the rate-limiting step in the cholesterol side, if we're going to regulate it, is the cytochrome P450 side chain, that cleavage enzyme complex in the first step. And so that's an important regulatory mechanism. We're talking about some of the hormones of the testes, which include testosterone and dihydroxytestosterone and 17-beta estradiol, which is a all these are important sex hormones. So these are the molecules that are produced there. So follicle-stimulating hormone binds to Sertoli cells and stimulates the synthesis of these and allows for the development of sex traits through these hormones. Estrogen, again, is synthesized through these, estradiol and estrone. So estrogen are formed by the aromatization of androgens through aromatase and ketoreductases, and so that's basically how those estrogens are formed. Steroids float around in the blood. They're lipophilic molecules, so they can transfer through the membranes, but they're also, since they're very hydrophobic, they need to be bound to protein carriers in the blood, and some common protein carriers are albumin, corticosteroid binding globulin, CBPG, or transportin. So our sex hormone binding globulins and our androgen binding proteins. So only the free fraction is biologically active, and that's only a small number, like less than 10%. And so this is how hormones get through the world. Hormones have a very short half-life. Steroids and thyroid hormones do have a long one, but the rest of them are seconds to minutes. And so we're going to see, you'll see that those have a very, very short half-life. Adrenal steroids in the blood, cortisol 70% is bound to corticosteroid binding globulin, CPG or CBG. 22% is bound to albumin and 8% is free. Aldosterone has 60% bound to albumin and 10% is to transportin. And that's basically how we get it from one location to another. For those two. For testosterone and estradiol, we have sex hormone binding globulin, SHBG, and progesterone binds to transportin. And all of these things at puberty, there's a small decrease in the level of SHBG in females and a large decrease in males, and that allows for a large biologically active population of sex hormones, and that allows for development to occur. So, as promised, we're going to talk about prostaglandins and some of the related other hormones. Prostaglandins also are related to thromboxanes and echinosides. They're very, very potent cell signaling molecules. They're hormones. They also have multiple effects, including the increase inflammation, and they also can have bad side effects that are associated with that inflammation. So they're synthesized from dietary fatty acids, in particular from arachidonic acid, as we mentioned yesterday. Here's some, as in the fatty acid lecture, here's some examples of their structures. You'll note that they're long-chain fatty acids, and then there's modified functional groups on there that allow them to perform additional functionalities. There's a lot of similarity in that they're long-chain hydrocarbons. So functions, we obviously have activation of the inflammatory response. They're also associated with blood clots to fix the damage, and some also have the opposite effect on reducing clotting. Certain prostaglandins involved with the induction of labor. They're involved with regulating all kinds of pathways, including salt, blood flow, mucus, acid synthesis in the GI tract, and there's some asthma medication associated with this. In particular, the inflammatory role is the one that most people are familiar with because of the COX-2 enzyme, which is one of the enzymes that's associated with the manufacturing. We'll talk about that here in a little bit. But COX-2 is the enzyme, and you'll get inflammatory prostaglandins. That's going to increase your inflammation, which also could increase pain. So if we inhibit COX-2, we decrease inflammation, decrease pain, decrease swelling. And that it kind of gives an idea of what the immune system response is. These hormones can also cause a significant upregulation in B cells, certain types of T cells, B cells, macrophages, and dendritic cells. So it's going to activate a whole range of cells in the immune system. The first step in synthesis is from dieteric linoleic acid. We're going to get elongation and desaturation. That will then generate arachidonic acid. From arachidonic acid, here's where we have our COX enzymes, which are cyclooxygenases, which basically create a ring structure, and that's generating the structure. That and peroxidases generate the structure of these sex hormones. So there's two catalytic activities here. We have our cyclooxygenase, our COX, and then we have our peroxidase. And that's going to allow us to generate a lot of these. Most of these hormones are generated that way. And this pathway is very emblematic of the whole process. I'm not going to go through all of them. I think that's unnecessary. All the prostaglandins just gives you an idea. And I think the important part is we have a drug target in the middle of this, which is the cyclooxygenase. There are two isoforms of COX. Both produce prostaglandins. COX-1 is constitutively expressed in most tissues. It's physiologically and homeostatic roles for cell signaling. It's not influenced by steroid administration and is not increased by cytokines or bacteria. COX-2 is inducible following inflammation and trauma. It's found in immunocompetent cells. So COX-2 is induced by cytokines and it maintains inflammation. And it can be inhibited by steroids, and it can also be inhibited by some of our common drugs. So prostaglandins play a key role in a lot of these different diseases, including cancer. And so they're very, very important drug targets, and you're likely to see a lot more activity associated with this. Although there was quite a heavy activity, in the 90s on this as a drug target. So, but this will come back. Also associated with periodontal disease. So the inhibitors, some of these are pretty famous: Vioxx and Celebrex, aspirin. And all the NSAIDs. They reduce production of prostaglandins and thromboxanes. They reduce pain, fever, and inflammation. Side effects: gastrointestinal irritation, ulcers, bleeding. So there's been a huge hunt associated with trying to find better NSAIDs. So aspirin works on both COX-1 and COX-2. Salicylic acid inhibits arachidonic's entry into the active site of the COX, and the acyl group of aspirin binds the serine in COX, and that's going to block the enzyme. It's non-selective and has multiple side effects. Tylenol inhibits the activity of COX-3, and that's going to obviously have different effects. So Tylenol is thought to be relatively more safe than some of the COX-1 and COX-2 inhibitors. Aspirin irreversibly inhibits COX-1 and COX-2, has a short half-life, and it will. So if we have low-dose aspirin, it will reduce but not abolish the ability to clot, and that will reduce the ability to have heart attacks and strokes in patients. So there's a lot of us. I take low-dose aspirin. There's a lot of us who do for that reason. All right. One other quick note. I was talking about gamma hydroxy. See if I can find this hydroxybutyric acid yesterday in class. See if I can find this lecture slide real quick. And there was a note about what so this is known as GHB, and you're probably familiar with GHB. I had kind of forgotten or didn't associate the two. So GHB is also known as 4-hydroxybutanoic acid. It's a neurotransmitter and a very psychoactive drug because it's a precursor to GABA and glutamate and glycine, and so it can bind to the receptor. And so it's commonly used as a general anesthetic, and it's used to treat certain diseases like cataplexy, narcolepsy, alcoholism, but it's also known as the date rape drug. So there you go. That's the structure right there. That gamma hydroxybutyric acid is your date rape drug, and that's why it's potent and illegal. So now you know. Might be a good case study topic. So, all right, with that, we're done. With this, I will post up the test questions here shortly, and hopefully you have a great 4th of July week. And if you have questions, please email.